Yeah.
Yeah. So I'm delighted to introduce Dr. Jody. Jody is from the University of Queensland. That's we do a significant amount of collaboration. She specialises in mosquito-borne virus discovery, the development of novel vaccine and diagnostic platforms. She's got 61 publications to date with amazing titles such as Nature Microbiology and Nature Communication. And she started her career working in industry at Aiken Biomedical, where she develops in vitro diagnostics tests, called pregnancy test strips and real-time PCR. She now has expertise in the development of monoclonal antibodies and has developed a whole suite of monoclonals that are really important in research for other viruses, as well as some of them are also being used commercially. And she's also been working to detect emerging viruses, and the work has led to a greater understanding of the mosquito virome. And she's also discovered these really novel insect-specific flaviviruses. We're going to talk to more about today, and how they've been turned into a for a vaccine. So thanks very much, Jody, for accepting our invitation, and we really look forward to hearing your thoughts.
Okay, thank you, Jo, for the introduction, and thank you for the opportunity to speak. I'm just going to share my screen. Hang on, screen one.
We can see it right.
Is that the correct screen? Yep. Okay, so
Jody, we are seeing the presenters view. You might want to do it in full view.
how do I do that?
Up in the display settings, you're in the drop-down menu. Yep,
Yeah.
and into view.
Okay, does that look right?
Yes, thank you.
Yep. Okay, so today I just wanted to give a bit of a snapshot of some really cool work that we have been doing with commensal viruses of mosquitoes. And these viruses have been found all over the world in mosquito populations and can only infect mosquitoes and don't replicate in vertebrates. And so our group has been interested in these insect-specific viruses now for about 10 years. And I guess we've got to the pointy part of our research where we can really start to exploit these viruses to our advantage. So I'm just going to take these off. And so the viruses that we have been focusing on are the insect-specific flaviviruses. And the story that I'm going to tell today is how we've been able to harness the benign characteristics of these insect-specific flaviviruses and engineer them into novel vaccine and diagnostic antigens. Now, this work has been highly collaborative, not only with many laboratories within the University of Queensland, but also QAMR Berghofer and also Queensland Health as well. And we're very thankful to quite a large team of enthusiastic and talented honours, masters and PhD students that have come through our lab over many years and have obviously contributed largely to this work as well. And right from the outset, I really wanted to acknowledge my boss, mentor and friend, Professor Roy Hall, just in case I ran out of time at the end of this presentation. And so back in 2010, he really went out on a limb to start this whole virus discovery program. So the viruses at the centre of my talk today are the flaviviruses, and significant members of this family obviously include the dengue viruses and also Zika virus, which really came into the limelight back in 2016 with the explosive outbreak in Brazil. What I really wanted to highlight on this slide was the transmission cycle of these pathogenic or vertebrate infecting flaviviruses. And they maintain a transmission cycle which involves an arthropod vector, for instance, a mosquito, or sometimes it may also be a tick. And then there's also the vertebrate amplifying host. And for some of these viruses, for instance, dengue, it may be humans or primates, other primates, or for West Nile virus, for instance, it would be birds. But these vertebrate infecting flaviviruses must be capable of replicating in both the arthropod and also the vertebrate. But the viruses that I am focusing on today are the insect-specific flaviviruses, and these viruses are maintained within a transmission cycle without the requirement for a vertebrate infecting, without a vertebrate host, and they are completely unable to replicate in vertebrate cells. So in the absence of a vertebrate amplifying host, these viruses are transmitted via a very strong mechanism of vertical transmission, which means that they are transmitted from the mother mosquito to her progeny, most likely via an infected egg. So as I said, we've been interested in discovering these viruses for about 10 years now and have looked in mosquito populations all over Australia. There are two main lineages of these viruses, lineage 1 and lineage 2. The lineage 1 ISFs are those that are most disparate genetically to the vertebrate infecting flaviviruses. And as we are seeing that this lineage is fleshing out with more and more of these viruses being discovered, they are now separating out into discrete clades. So we've got those that are associated with Anopheles mosquitoes, those with Aedes, and also those associated with Culex mosquitoes. The lineage 2 ISFs, on the other hand, are much more genetically closely related to our vertebrate infecting flaviviruses down here. And to date, not as many of these lineage 2 ISFs have been discovered. So back in 2010, the first ISF we discovered was Palm Creek virus, and this was from mosquitoes collected in the Northern Territory and also northern regions of Western Australia. Subsequent to this, we isolated Parramatta River virus from Aedes vigilax mosquitoes collected around Sydney, and we've also found it in the same mosquito species as far north as Brisbane. Self-fusing agent virus has been isolated from Aedes aegypti mosquitoes from northern regions of Queensland. And we've also detected a whole new group of new flavivirus species from Anopheles mosquitoes. And the virus that we've done the most research into is Corumba virus. So all of these viruses that I've just mentioned here all belong to lineage 1. It actually took us a very long time to finally get an isolate of a lineage 2 ISF, and this was Binjari virus. And you will see as I get further into my talk why this virus has become so special to us. Now, we knew this virus existed because back in 2010, we did detect its genetic RNA in a mosquito pool that was collected from Katherine in the Northern Territory. But we could not isolate a virus from it because that mosquito pool had not been kept on the cold chain sufficiently. We actively searched for this virus for 6 long years, and it wasn't until the end of 2016 that we finally got an isolate, and this was very much a eureka moment for the lab. And that isolate came from another region within the Northern Territory. So as fate should have it, however, very soon after finally isolating Binjari virus, we isolated another very close cousin to Binjari, which is a virus that we've named Hidden Valley. And a lot of this work was done by Dr. Jessica Harrison as part of her PhD in our lab. So we've got quite a large suite of these insect-specific flaviviruses now, and what I really wanted to highlight here is that consistent with other reports of both lineage 1 and lineage 2 ISFs that have been detected all over the world, none of these replicate in vertebrate cells, and this is only a very small snapshot of a very extensive panel of vertebrate cell lines that we've assessed these viruses in. And we have concluded that, yes, they don't replicate in any of these cell lines. So now that we had this panel of ISFs, we asked the question, could we engineer these viruses and really harness the benign nature of these viruses such that we could make recombinant vaccines and diagnostics? So utilize the replication machinery of the insect-specific flavivirus, but make it look like the pathogenic virus on the outside of the virus particle. And so one of my colleagues coined the phrase that perhaps we should be calling these sheep in wolf's clothing. So something that was very benign and safe on the inside, but looked very scary on the outside. So the way that we went about this was to take the genetic backbone of the insect-specific flavivirus, so all of the replication machinery, and to exchange the PRMN structural genes, so these are the proteins that form the outside of the virion, exchange the ISF ones with those of a pathogenic virus. So for instance, West Nile virus. To do this, we used a method called the circular polymerase extension reaction. And this has really been brought into the field of flavivirology by Professor Alex Cromick and Dr. Yin Sito, who we work very closely with. And this technique has really revolutionized the way that we can prepare these chimeric particles. So this procedure involves us amplifying up our insect-specific flavivirus backbone using overlapping PCR products, and also amplifying the PRME genes of our pathogenic virus, whether it be dengue, Zika, West Nile virus. These are then connected with a linker region, which contains our insect promoter to drive the transcription of the RNA in mosquito cells. All of these PCR products are put into a tube. We do a few PCR cycles, and this whole mixture is then transfected into our mosquito cells. We allow them to incubate for about 7 days, after which we will passage the supernatant maybe 1 or 2 times. What has been really great is that we've got this system working so efficiently now that even if we have to order any of these fragments as gene blocks, we can produce new chimeric viruses within 2 to 3 weeks. So it is, it's very good in terms of I guess, disease outbreak response. So initially on paper, I guess we thought, well, this was going to be incredibly easy. Yeah, we'll just exchange these genes. But in practice, it was not. And we actually burnt through quite a number of our ISFs trying to find a suitable partner to chimerize. So Corumba virus, cell-fusing agent virus, Parramatta River virus, and we tried it extensively with Parramatta River virus. None of these viruses could be successfully chimerized. And it wasn't until we finally went back to our prototype virus, which was Palm Creek, that we finally had success. And this probably took a good 18 months, I think, and one very poor PhD student. So this staining here, the green staining, is showing a successful chimera that has the Palm Creek virus genetic backbone and the structural proteins of West Nile virus on the outside of the virion. Since that time, we've now produced a number of these Palm Creek virus backbone chimeras. So we've got the West Nile virus one, Zika, and also dengue. And we were pleasantly surprised to see that when we did growth kinetics on these chimeric particles, that they actually replicated really well and often outgrew their wild-type pathogenic counterparts. A lot of you may be surprised that often in cell culture, flaviviruses don't always replicate that well, and dengue virus is a key example of this. And so it was great to see that our PCV dengue chimera was outperforming wild-type dengue by about a log. And similarly, with our PCV Zika chimera, it was also growing really well as well. We were, however, a little bit disappointed with our West Nile virus chimera and wondered if we could do better. And by this point in time, we had finally got our isolative Binjari virus. And we knew that this virus had some very unique characteristics. First of all, despite the fact that it is a lineage 2 ISF, it did not replicate in any of the vertebrate cell lines that we had tested on, which was very good news. It also has incredibly high replication in mosquito cells. So we're looking at almost 10 to the 10 infectious units per mil, which is very high for a flavivirus. And it also exhibited very high tolerance for the exchange of its pyrimidine E structural genes. And just to highlight this, on this phylogenetic tree here, every virus here where I've got a little star, we've successfully chimerized. So we've got, I think at last count, probably between 40 to 50 different Binjari virus chimeras now sitting in our freezer. What was even more exciting is the replication of these chimeric particles. So I don't know why that keeps popping up. And we saw that particularly with our Binjari Zika chimera, its replication is extraordinary. So we're seeing 10 to the 10 infectious units per mil, by far outcompeting the wild type Zika. Dengue, we're seeing that our chimeric particles are replicating more than 1,000 times better than wild type dengue. And so as you can imagine, if you're trying to produce a vaccine or a diagnostic antigen, having something that replicates so well is very attractive for commercialization. And just to highlight how much better Binjari is as a backbone than, say, PCV, we have done some direct comparisons. And so this is the growth here of our PCV Zika chimera. And we're seeing that our Binjari Zika chimera is outperforming about 2 logs. We've also done some very crude estimation on how much purified antigen we're getting from these chimeras. And I've seen that with our West Nile chimera that we can purify up to 7 mg per litre of our West Nile virus antigen. And this is either comparable or exceeds reports that I've seen in literature for other virus-like particles. And then importantly, we needed to assess whether these chimeric particles were safe to use. Could they replicate in vertebrate cells? And you need to understand now that these particles looked like the vertebrate infecting flavivirus on the outside, we're presuming that they can actually bind and enter the vertebrate cell. But we were very happy to see that none of these chimeric particles, and we've assessed all of these now, none of them are capable of replicating in vertebrate cells. I'm just going to touch on this a little more because we have done a lot of extensive work looking at the safety of these particles. And so, as I said, one of the blocks that we have removed for replication in vertebrate cells is that of cell entry, because these particles do look like the vertebrate infecting flavivirus on the outside of the virion. So we are presuming that they are binding and entering this vertebrate cell. But once inside, they have to, there are a lot of other potential barriers to their replication as well. Flaviviruses have to interact with host factors at every step of the way during their replication cycle. And so perhaps these insect-specific flavivirus, their replication machinery may not be able to interact with the vertebrate cell host factors. Temperature may also be an issue. As you could imagine, these insect-specific flaviviruses have evolved in the mosquito and have not had to deal with going into a mammalian system at 37 degrees. Vertebrate cells also have very sophisticated antiviral responses, for instance, interferon, and the vertebrate infecting flaviviruses have developed very unique mechanisms in being able to either evade or suppress some of these antiviral responses. And perhaps the ISFs have not developed the ability to be able to evade these responses. And there also may be issues with the exit of these particles as well. So just to very quickly look at some of the work that we've done, we've basically tried to remove as many of these particular barriers as possible. So we've removed the cell entry barrier because these particles look like the vertebrate infecting flavivirus. We've also used cells that are deficient or have a defective interferon response. And the cells that we've used here are these MEF-IFNA knockout cells. And simultaneously, we've also reduced the temperature down to 34 degrees. But despite doing this with our chimeric particles and removing as many of these barriers as we can, we are still not seeing any replication. And so this tells us that with ISFs, there are definitely, there is a post-entry block in the absence of an intact interferon response and lower temperature. But of course, just because we have defined something in vitro doesn't mean that it necessarily is the same in vivo. And I'm not going to go into this graph, but I just wanted to highlight that we've also put a very high dose of our Binjari Zika chimeric particles into severely immunodeficient mice, and have demonstrated that these particles have no detectable capacity to expand in mammalian tissue in vivo, which means to us that they are safe. They do exhibit a very high safety profile. So I guess just quickly on to what we're now doing with these chimeric particles, and some of the really exciting work that's coming out of our lab and our collaborator, Dr. Dan Waterson, is some of the structural work that we're doing. And what he has been able to show through some very exquisite cryo-electron microscopy is that he can reconstruct these particles. And this is our Binjari Zika particle here. This is a reconstruction down to 7.1 angstroms. And by doing this, he has shown that our Binjari Zika particles are structurally indistinguishable from wild-type Zika. We have also got an active collaboration with Monash University, and they have taken wild-type West Nile virus and also our Binjari West Nile virus and have imaged them simultaneously, and they're now down to, I think it's less than 4 angstroms, and have shown that these particles certainly are structurally indistinguishable. And this, what I really need to highlight here is this high level structural authenticity is not typical for standard recombinant virus-like particles. And what this means is that we now have the ability to start solving the structures for some of the more pathogenic flaviviruses for which there are no structures already out there. So it's really exciting times for us. So just on to the vaccine applications. Today I'm going to just step you very quickly through the Binjari Zika studies that we've done, but we just recently had published our work looking at the efficacy of our Binjari West Nile virus chimeras against pathogenic West Nile virus strains. We've got a Binjari yellow fever vaccine paper that will probably be published within the next few weeks, and a Binjari dengue paper that's about to be submitted, so watch this space. So the work with our Binjari Zika chimera was done in the lab of Professor Andreas Subia at QAMR Berghofer, and he used some non-lethal IFNA knockout mouse models, one of these being the male testes damage model, and that's the data that I'm going to show today. And what he showed was that a single unadjuvanted dose of only 2 micrograms, so we're talking a really small dose here, that these particles provided protection in this male mouse model. So after injection of our Binjari chimeric particles, we looked at the antibody responses and saw that these mice did develop a very robust immune response against Zika virus, and also a robust neutralizing antibody response as well. Andreas's team then went on to challenge these mice with Zika and saw that our chimera vaccinated mice did not have any circulating virus within their bloodstream, so no viremia. And this is in comparison to quite high levels of viremia in the PBS control mice. So as I said, this is a testes damage model, and so what happens in the male mice when they are infected with Zika is they get overt hemorrhage and also shrinkage of the testes. And so this is the PBS control group here. But as you can see, with those mice that received the Binjari Zika vaccine, there was no overt damage observed. And this was also seen when the weights of the testes were performed as well. So significantly lower weight of the testes of the mice that received no vaccine as compared to those that were vaccinated. And here's just some beautiful IHC staining. So all of the staining on the right-hand side here is in the PBS group, and we're staining for Zika antigen, non-structural 1 protein antigen within those testes, whereas in the vaccinated mice testes, there was no staining for Zika virus antigen present. So onto the diagnostic applications. Initially, we confirmed that these particles were antigenically authentic, and this was performed using a very large suite of monoclonal antibodies that we have in our laboratory. And these target all three domains of the envelope protein and also the PRM protein as well. And so this graph here is just showing that we have got very high correlation between the binding of these antibodies to our chimeric particles as opposed to wild type antigen. And I guess you would expect that since we now know that structurally these particles are so similar as well. We've also used them in multiple different diagnostic assays. The first of these was an ELISA. And what we found was that these particles are secreted at such high concentrations in the culture supernatant of mosquito cells that we can actually use the culture supernatant directly from the flask and capture these particles using an antibody that's coated on the ELISA plate. And then we can bring in our human serum on top of this. And so these graphs here just demonstrate that we were able to accurately diagnose patients that have been infected either with West Nile virus or with dengue virus as well. And we've also demonstrated this for Zika infected patients as well. We've also done a lot of work with Dr. David Warrilow up at Public Health Virology at Queensland Health, and they have generated a really neat microsphere immunoassay. And this assay they use for their routine diagnosis of flaviviral diseases. And what they showed was that our chimeric particles could be used in their microsphere immunoassay and exceeded their expectations so much that they're actually now rolling out this technology into their laboratory. And I guess the reason for this is that they no longer have to produce large quantities of pathogenic flaviviruses such as Japanese encephalitis virus. They can now use these much safer chimeric particles to generate their diagnostic antigens. And I guess the future work that we're now doing, and this is where we are now collaborating with Joanne's group, is that we are now starting to apply these chimeric particles to lateral flow assays, so these point of care assays. And I guess this harks back now to where I began in industry, so I've kind of come full circle. So we're hoping to generate these lateral flow devices not only for human health, but also for veterinary applications as well, because animals do become ill. For instance, horses can become infected with West Nile virus and Murray Valley encephalitis virus. So to be able to test animals will be very handy as well. And of course, these devices are very cheap to manufacture. So just in summary, we have discovered a very genetically diverse group of ISFs in the Australian mosquito populations. And we've found that they do provide a great vehicle for the production of safe, antigenically authentic vaccines and diagnostic antigens. Importantly, we can grow these and use these under minimal biocontainment. And so this means that in the future, we can produce antigens for also BSL-3 and 4 pathogens. And with this 2 to 3 week turnaround, this technology is really ideal for new disease outbreaks. So as I mentioned, a very large team have contributed to this work. This is not everyone that's been involved in the whole mosquito collection program as well. But yeah, I do want to thank everyone that's been involved and also our funding agencies. Thank you.
Thank you very much. That is fascinating work. It's lovely to hear that such amazing work is collaborating with Joanne. I'm very proud of her, of course. I can see that there's a couple of questions in the chat. Are there any questions for Jody from the audience?
I'm happy to start. Can you hear me?
Yes, I can.
Excellent, cool. Have you considered trying to put other proteins into your insect-specific flavivirus sort of structure? I mean, why just stick to arboviruses?
Yeah, we have considered that. I mean, we've even considered other arboviruses. I don't know how easy it will work, but yeah, one of our collaborators has had some ideas. But I think the strength of the technology really is the fact that we can that these proteins assemble so authentically. So by putting other proteins on there, I'm not sure how successful the replication of those resulting viruses would be.
That makes sense, I guess, because it's the polyprotein that gets processed up. If other proteins aren't used to being treated that way, they might not work as you'd expect.
Jody, there's a few questions here from Samuel Phillips for you. I don't know if you can read in the chat and answer directly.
Are there plans to include adjuvants in the vaccine makeup? So we did actually test these chimeric particles with an adjuvant as well, and found that there was no enhancement in the ability of these particles to protect. So at this point in time, we're hoping to use it without an adjuvant, but it also comes down to minimizing the dose. So I guess it's weighing up the amount of the protein that needs to go in versus whether it might be better to also have an adjuvant. So how long was the IgG antibody response? We have assessed mice up to 6 months and saw that they still had a robust immune response. Is there any work looking at the cellular immune response? We are hoping to do that in the future, but we have not done that yet.
I have a question. It's and obviously all the diseases or viruses that you talked about have horrific effects, and Zika is particularly one that people just have a very emotive response to.
Yeah.
How long before a vaccine is available for people?
I guess in terms of Zika, I mean, there's a lot of vaccines already out there in the last stages of clinical trials, and those vaccines probably won't be released until there's another outbreak. In terms of our work, our biggest hurdle at the moment is that we will need a GMP approved mosquito cell line. So that is our biggest hurdle, and that is something that we are currently working on.
Any other questions for Jody? No? Okay. Well, in that case, Jody, thank you so much for sharing with us your incredible work. Thank you, Joanne, for recommending such a wonderful speaker. And we really appreciate your time and actually coming and listening to us as well. It's really much appreciated.
Okay. Thank you for the invitation.